Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are working our way through Chapter 5, and we are talking about cellular respiration. We just had the last video, which was chemiosmosis, and that showed you the very end step at this process, but now we need to go back to the beginning of cellular respiration and see what happens to get to where we're making phosphorylating adenosine diphosphate to make adenosine triphosphate. Okay, so overview of cellular respiration. Cellular respiration involves taking glucose, C6H12O6, adding some oxygen, adding some water. This process requires water. And then after that, you are going to make energy. Energy is what you're trying to make in respiration. Remember, without ATP, we die. We have to make energy. Now, there are some other things that get made during this process. We make some carbon dioxide, we make some water. We weren't trying to, so carbon dioxide and water are considered byproducts of cellular respiration. ATP is our end product. That's what we're trying to make. Glucose is our precursor, what we're starting with, and then there's other precursors that are needed to make this reaction happen. Now, reactions, just like math, are usually very careful. The number of carbons on this side has to equal the number of carbons on this side. But you see there's a little bit of fudgy math here, 36 or 38, and that's because the process of chemiosmosis is not exact. And so there's a little bit of fluctuations on how many ATP you make per molecule of glucose. Now we are going to keep count, but do know it's not an exact science, but it is an approximate and it's an average from that. So for every molecule of glucose we have, we can produce 36 or 38 ATP. All right, so what's the difference? You may have heard 36 ATP, maybe in some other classes. Bacteria, prokaryotic cells, can actually produce 38 ATP. It's a little bit more efficient, gets a little bit more energy made per molecule of glucose. And we'll talk about why as we go through. Okay, so for prokaryotes, your number is about 38 ATP. All right, so why do we need to make that energy? I told you that, you can't take a banana and put it on your arm and expect arm day to work out for you. Okay, so the cell, it's kind of like a currency. If you walked into McDonald's with a $100 bill, they're going to laugh at you. You're not going to get a Big Mac. You're not going to be able to feed yourself. Even though a $100 bill is currency, it's a larger denomination than McDonald's can accept. Well, glucose, when we get glucose, even once we break it down out of the banana, we get glucose, that's still a large denomination. We need to break it down into a currency that our cells can accept. So each glucose has a lot of energy locked up into it, just like your $100 bill. But in order to get it, to get the food, to get the energy out of it, we're going to have to break it down into currency that they'll accept, that the vending machine will accept, or McDonald's will accept. So glucose has energy, the cells can't use it until it gets broken down. So that's what you're doing. We've already talked about the byproducts of this reaction. So let's write down the steps and keep those kind of handy as we're going through the video to refer to them. I usually write them on the board in class. So this process is a pretty long process, and it involves several metabolic pathways. So the several metabolic pathways it involves all have their own names. So the first stage of cellular respiration is a metabolic pathway called glycolysis. Now in that glycolysis, which actually stands for glyco as sugar and lysis, we know lysis is to break apart. So we're actually breaking apart sugar. You're going to start with the glucose and you're going to break it down some. We do make a little bit of ATP, but not much in that. But we do make an end product that can go through a couple of reactions called the prep steps or lysis of acetyl-CoA to produce acetyl-CoA, and that will be the precursor for the Krebs cycle. Krebs cycle, we're still going to make a little bit of ATP there, but we're going to make a lot of molecules that are going to be necessary for chemiosmosis. Chemiosmosis is the last process, and chemiosmosis occurs at a place called the electron transport chain. Now that electron transport chain is back to what I showed you on the very beginning, which was the cell membrane with those integral proteins and the hydrogen ions, this whole place is going to be called the electron transport chain. The process that occurs is called chemiosmosis. Okay, so keep those in your mind. Electron transport chain is the place this occurs, and then the process of making ADP phosphorylating ADP to make ATP is actually called chemiosmosis. Okay, so let's get started. This is another overview kind of slide, picture slide. We're going to come back to this. Respiration, cellular respiration. Now, not respiration like breathing in and out. This is completely different. It's cellular respiration. Starts with the first stage, which is glycolysis, where we take glucose as our precursor and we end up making pyruvic acid. We can continue on to make acetyl-CoA, enter the Krebs cycle, and then chemiosmosis at the electron transport chain. This is the process of cellular respiration, the process of glucose catabolism. We're breaking down glucose to make energy and releasing energy from that. All right, so we'll keep coming back to this. Okay, so just to kind of help you put down layers, because you've learned some of this in the past, in a prokaryotic cell, the first three stages, glycolysis, did you write them down? Glycolysis, the prep steps, and then the Krebs cycle have to occur in the cytoplasm. They don't need a cell membrane for any of that, and there's no place else in a prokaryotic cell for these things to happen. The electron transport chain needs a membrane. So the only membrane, if you remember, in a prokaryotic cell is your cell membrane. Now, some prokaryotic cells, the gram-negative ones, have an outer membrane, but not all. So the outer membrane is not used for this. This process occurs in the cell membrane of a prokaryotic cell. Just so you can, again, coordinate this with previous knowledge and future knowledge, eukaryotic cells, this is occurring in the mitochondria. So glycolysis occurs in the cytoplasm, but the prep steps in the Krebs cycle occurs in the mitochondria. Now, not in the membranes of the mitochondria, it actually occurs in the middle, which is referred to as the matrix of the mitochondria. And then the electron transport chain is located in that cristae of the mitochondria. All right, these figures, you do not need to know all the little bitty names and stuff in these figures. It was just a good kind of visualization of where this process is occurring. You can see this electron transport chain. You can see the ATP synthase. This is happening in the prokaryotic cell membrane versus this is, in the mitochondria. You can see the cristae, the matrix in the middle, where the Krebs cycle is occurring, and then the cristae, the inner membranes of that. So it's just a good comparison slide for the locations of this. All right, so let's do glycolysis.